Welcome to Sciencera. In this video, we're going to discuss major histocompatibility complex MHC1 and MHC2. Let's begin. Introduction. The genetic capability to mount an immune response is linked to a group of molecules originally referred to as the human leukocyte antigen. French scientists gave them this name because they were first defined by discovering an antibody response to circulating white blood cells. These antigens are also known as MHC because they determine whether transplanted tissue is histocompatible and thus accepted or recognized as foreign and rejected. MHC molecules are found on all nucleated cells in the body, and they play a pivotal role in development of both humoral and cellular immunity. Gene coding for MHC molecules in human are found in short arm of the chromosome 6, inherited as autosomal dominant gene, inherited both genes of the both parent. Let's look at the gene coding for MHC molecules. Gene coding for MHC molecules in humans are found on the short arm of chromosome 6 and are divided into three classes. Class 1 molecules contain HLA A, B, C. Non-classical components contain HLA E, F, and G. These are coded for at least three different locations or loci. Class 2 molecules genes are situated in the D region and therefore are several loci DQ, DR, and DP. Non-classical HLA DM, HLA DN, and HLA DO molecules. Lastly, class 3 molecules genes are located in between class 1 and class 2 regions to produce complementary proteins C2, C4, and factor B. Production of cytokines such as TNF involves in antigen recognition. At each of these loci or the location, there is a possibility of multiple alleles. Alleles are alternate form of gene that code for slightly different varieties of the same product. The MHC system is described as polymorphic because there are so many possible alleles at each location. So, part of class 1 molecule HLA-A has at least 580 different alleles, B has at least 921 different alleles, and C has at least 312 different alleles. Let's look at the diagram of MHC regions of human chromosome 6 and the gene product associated with each locus or the location. So, starting from the class 1, locus A, about 95% of allelic class 1 molecules. Locus C, from the class 1 molecule 50 allelic, one of the 50 allelic, and B, one of greater than 200 allelic class 1 molecules. Class 2 molecules locus are DR, DQ, and DP. On DR, the product is alpha and beta chains of class 2 molecules. DQ products are the various alpha and beta chains of class 2 molecule, and DP also various alpha and beta chain of class 2 molecules. Class 3 molecules products are locus or locations are C4, BF, and C2. C4 product is the fourth component of the complement system. BF product factor B of the complement system, and C2 is the second component of the complement system. So this is the major histocompatibility complex on chromosome 6. Gene coding for MHC molecules. The probability of that any two molecules will express the same MHC molecule is very low. An individual inherits two copies of chromosome 6, and thus there is a possibility of two different alleles for each gene on chromosomes unless that person is homozygous. MHC alleles are codominant, they are always expressed in the phenotypical level. Since the MHC gene are closely linked, they are inherited together as a package called haplotype. Thus, each inherited chromosome region consists of a package of gene A, B, C, D, R, D, P, and D, Q. The full genotype would consist of two of each gene at a particular locus. So, for example, in the table you can see A19 and A16s. A16 on the complexity complex, histocompatibility complex. HLA, A16 and 19, both gene will be present on a particular locus on the genotype. The structure of MHC1 molecule. Each of MHC gene codes for a protein product that appear on cell surface. All the proteins of a particular class share structural similarities and are found on the same type of cell. Class 1 MHC molecule expressed on all nucleated cells, alpha folded into three domains A1, A2, A3 planted on the cell membrane as you can see on the diagram. The A3 region react with CD8 on cytotoxic T cell. B2 microglobulin does not penetrate the cell membrane, but it is essential for proper folding of a chain. Then we have a structure of MHC2 molecule. The occurrence of class 2 MHC molecule is much more restricted than that of class 1. The major class 2 molecules DP, DQ, and D are more polymorphic with 18 different alleles, consist of two non-covalently bonded polypeptide chain that are both encoded by the gene in MHC complex. DR is expressed at the highest level as it accounts for about one half of all of the class 2 molecules on the particular cell. Other class 2 genes have been described as DM, DN, and DO. These are the non-classical genes. These are found on B cells, monocytes, macrophage, and the dendritic cells. Antigen presentation. The main role of the class 1 and class 2 MHC molecule is to bind peptide within cell and transport them to the plasma membrane, where T cell can recognize them in the phenomena known as antigen presentation. Class 1 molecule mainly present peptide that have been synthesized within the cell to the CD8 plus receptor or T cytotoxic cell. Class 2 molecules mainly bind to exogenous proteins and present it to the CD4 plus or T helper cell. It is thought that two main classes of these molecules have evolved to deal with two types of infectious agent. Class 1 process those infectious agents that attack from inside, and class 2 process those infectious agents that attack from the outside. Role of class 1 molecule. Both MHC class 1 and 2 are synthesized in the rough ER. Connexin, it keeps the alpha chain in partially folded state while it awaits binding to the beta 2 macroglobulin. Three other chaperone molecules, calreticulin, tapicin, and ERP57, are associated with complex and help to stabilize it for the peptide binding. Class 1 HLA, these are effective for endogenous antigens such as tumor, virus, and parasite. Class 2 HLA effective for exogenous antigen such as bacteria. Class 1 HLA molecules are presented in CD8 plus or T cytotoxic cells, while class 2 is presented in CD4 plus or T helper cells. Class 1 HLA proteasomes or are the cylindrical contain enzyme that process antigen. Transporting peptides includes TAP1 and TAP2. In class 2, HLA invariant chain maintain the class 2 not yet accommodated within antigen invariant chain peptides are present. Let's see antigen presentation by MHC1. Digestion of these defectives or early protein is carried out by the protease that reside in large cylindrical cytoplasmic complex called proteasomes. Proteasomes are pack of enzymes that play a major role in antigen presentation. Once cleaved, the peptide must then be pumped into the lumen of endoplasmic reticulum by specialized transporter protein. These protein transporters associated with antigen processing TAP1 and TAP2 deliver the process antigen to ER where MHC molecules from the cytoplasm to the lumen of ER of the peptide suitable for the binding to class 1 molecule. Chapecin, which helps TAP transporter to have proximity to the newly formed MHC class 1 molecules and mediates interaction with them so that peptide can be loaded onto the class 1 molecules. Once the alpha chain has bound the peptide, the MHC1 peptide complex is rapidly transported to the cell surface. Let's look at the role of class 2 molecules now. Unlike class 1 molecule, class 2 molecules must be transported from the endoplasmic reticulum to an endosomal compartment before they can bind peptides. Dendritic cells are the most potent activators of T cells and they are excellent at capturing and digesting exogenous antigen such as bacteria. Invariant chain prevent interaction of the binding site with an endogenous peptide in endoplasmic reticulum. Invariant chains serve to protect the binding site. Once bound to the invariant chain, the class 2 molecule is transported to an endosomal compartment where it encounters peptide derived from the endocytosed exogenous proteins. The invariant chain is degraded by protease, leaving just a small fragment class 2 invariant chain peptide, also called CLIP, attached to the peptide binding cleft. CLIP is then exchanged for the exogenous peptide. Let's look at the summary for recognition of foreignness. MHC molecules are cell surface proteins coded by a group of genes termed as the major histocompatibility complex. Class 1 MHC proteins are found on all nucleated cells of the mammal, and class 2 MHC proteins are only expressed on cells that can phagocytose foreign materials and organisms. The human MHC gene product are called the human leukocyte antigens or HLA. Class 1 MHC proteins collect foreign peptide processed by the proteasome and present them to the cytotoxic cells, and class 2 MHC protein collect foreign peptide processed by the phagosome and present them to the helper T cell. This brings us to the end of today's video. I hope you understand the topic. If you like my video, don't forget to subscribe for more.